Have you ever been prescribed an antibiotic? Antibiotics are the medication that we use to kill bacteria that cause infections. But did you know that some bacteria can become resistant to them?
Hello, my name is Raquel Ferrer, and I am a postdoctoral researcher at Harvard Medical School, where I am studying bacterial resistance to antibiotics.
Bacteria are microscopic single-cell organisms that can sometimes be beneficial, like the ones that we use to make yogurt, and sometimes be harmful, like the ones that cause ear infections. One thing that they all have in common is that they carry genes on their inside. Genes are like the instruction manual that decides how the bacteria is going to look and act. Sometimes those instructions can include information about how to become resistant to an antibiotic, and this is what we call genotypic resistance. Bacteria have 4 types of genotypic resistance. Many antibiotics need to go inside the bacteria to effectively kill it. So the first way that bacteria have to become resistant to antibiotics is impermeability. Impermeability acts like a shield that prevents the antibiotic from going inside the bacteria. Other genes can carry the instructions about how to build pumps. Pumps are capable of kicking out the antibiotic once it has reached the inside of the bacteria. Another way to become resistant is that antibiotics need to bind to their targets like a key to a lock. So a very clever way that bacteria have found to become resistant to antibiotics is to change the lock so that the interaction is no longer possible. Finally, some bacteria have genes that tell them how to create an enzyme to cut the antibiotic molecule. This is the 4th mechanism of genotypic antibiotic resistance. But bacteria have another sneaky way of becoming resistant to antibiotics besides the one that is encoded on their genes. This kind of resistance is called phenotypic resistance, and is what my research is focused on. But what is phenotypic resistance, you might be wondering? Well, phenotype is the way a bacteria looks and acts based on both the genotype and the environment. I'll give you an example. Imagine a couple of identical twins. This means that their genotype is exactly the same. Their genes can say that they are both going to be thin. But what happens if one of them decides to go to the gym 5 days a week? Even if their genes say that they both have to be thin, the one that goes a lot to the gym will start to get stronger and stronger because the environment has affected the physical expression of his genotype. In the case of bacteria and their ability to become resistant to antibiotics, one example of phenotypic resistance are dormant bacteria. You can think about them as bacteria that are sleeping. So the same way that while you are sleeping you don't eat food, these bacteria do not take nutrients from the environment. And if the antibiotic is there, they will not take it in and they will survive the exposure. Genotypic antibiotic resistance is very well understood and is routinely studied in the clinic to decide the best antibiotic treatment option for the patient that is suffering from an infection. On the other hand, phenotypic antibiotic resistance is more unknown and is normally overlooked in the clinical setting. As a result, a patient might be prescribed an antibiotic to which the bacteria is not genotypically resistant, but is phenotypically resistant. This causes all the phenotypically susceptible bacteria to die from the antibiotic, and allows for the phenotypically resistant bacteria to take over the infection and cause a treatment failure. The reason behind genotypic resistance being better known and understood than phenotypic resistance is that phenotypic resistance is very difficult to study. In genotypic resistance, scientists can study bacteria as a group, because as opposed to humans, bacteria can have millions of twins. But in order to study phenotypic resistance, we need to see bacteria as individual cells, one by one, because the frequency of appearance of this resistance is very small. This has proven to be challenging because bacteria are tiny. As a reference, bacteria are 1,000 times smaller than a grain of sand. So what clever ways have scientists found to study bacteria one by one? In my lab, the approach that we use is fluorescence microscopy and microfluidics. Fluorescence microscopy allows us to see very small bacteria where we have used fluorescence to mark genes of interest. In microfluidics, we use a special chip with tiny places where we trap bacteria and study them as they divide. We can also add a nutritive soup that contains what bacteria need to grow and divide and can even add antibiotics to it. Using both microfluidics and fluorescence microscopy, we can take time-lapse images and see how bacteria behave under the same or different environments. First, we load our bacteria of interest in the microfluidic chip and centrifuge it so the bacteria are trapped in dead-end alleys. Then, we attach some tubing to the chip to be able to provide the bacteria with the nutritive soup that they need to divide. Finally, we put the chip under the microscope and observe the bacteria, taking images every certain amount of time. With these kinds of experiments, we can make videos like this one. In this video, we have loaded many fluorescent twins of the same bacteria to a microfluidic chip, and we have taken images of them for some time. Then we have added an antibiotic and we have seen that most of the bacteria die. You will see them lose fluorescence as they pop. Finally, we have removed the antibiotic and have allowed for the surviving bacteria to recover. Why did some bacteria survive the antibiotic treatment if they are all genetically identical to their siblings? This is the question that we are trying to answer in my research. Answering it will give us a better understanding about how phenotypic antibiotic resistance works, information that will be very helpful to prevent some antibiotic treatments from failing. I hope you have enjoyed learning about phenotypic antibiotic resistance as much as I have enjoyed making this video. Thanks for watching.